speaker is Inken, who's going to talk about zebrafish as a model of cardiac conduction defects.
Thank you. Okay. Yes, so the title of my talk is a transgenic zebrafish model of a human cardiac sodium channel mutation exhibits bradycardia, conduction system abnormalities, and early death. Now, as most of you will be aware, we're in the midst of a genetic revolution with the advances of next-generation sequencing generating huge amounts of new genetic variants. Now, determining which of those genetic variants are of functional significance and which ones are benign poses a significant challenge. We're in great need of finding time-efficient ways to evaluate genetic variants, and in vivo models, in particular the zebrafish, are gaining increasing popularity in that respect. So why are zebrafish so popular for the modeling of human diseases, in particular cardiovascular diseases? Well, zebrafish are very easy to maintain. They produce large numbers of offspring. As you can see here, they develop rapidly externally outside the womb and are optically transparent. That means the development of organs and the function of organs can be observed in real time just using a light microscope without needing any manipulation. The genome of the zebrafish is conserved and highly amenable to genetic manipulation. Furthermore, zebrafish embryos do not depend on a functioning circulatory system, as they can obtain oxygen via diffusion from the surrounding media. Lastly, zebrafish have a very similar heart rate, action potential, shape, and duration, and ECG morphology to humans. Now, the aim of my project was to generate a transgenic zebrafish model of an arrhythmia-associated variant in the SCN5A gene, which encodes the human cardiac sodium channel. Why did we do that? Because so far no other transgenic zebrafish models of human heart disease mutations exist in literature, and proof of principle is required that human cardiac phenotypes can be replicated in transgenic zebrafish. Now, we're hoping that our transgenic zebrafish model of the particular mutation that we're studying will enable the evaluation of pathophysiological mechanisms and the development and trial of new therapies. As I mentioned, SCN5A encodes the alpha subunit of the human cardiac sodium channel, NaV1.5. This channel carries a large inward depolarizing current, which underlies the initiation and propagation of the cardiac action potential. This current plays a pivotal role in cardiac excitation and conduction, which is reflected by the fact that SCN5A mutations are very prevalent in, patients with cardiac phenotypes very often have SCN5A mutations and show diverse cardiac phenotypes, as I've shown in the bottom, including very many arrhythmic phenotypes. Now, there's very robust evidence that the D1275N variant is indeed a disease-causing mutation. There's genetic evidence. It's a highly conserved residue, which has been independently identified in 4 large families. An example of one of those pedigrees I've shown you in the bottom right. And consistently, this variant is associated with cardiac phenotypes such as atrial standstill, sinus node dysfunction, arrhythmias, conduction system defects, and dilated cardiomyopathy. There's also robust functional evidence that this variant is disease-causing. For example, a knockout, knock-in mouse model was generated a few years ago. And as you can see on the left from those ECG traces, mice that homozygously express this variant show cardiac conduction disease, atrial fibrillation, and atrioventricular conduction block. So I've lost my mouse. Don't know where it's gone. Point to it, but I can't. Well, patch clamping of ventricular mouse cardiomyocytes, shown on the right, shows that there is significant changes in the sodium current in mice that express homozygously that variant. Perfect. There we are. Now, how did we go about generating transgenic zebrafish which express human SCN5A? we cloned an expression construct for TOL2-mediated transgenesis, which contained 2 genes in the one cassette. On the one hand, human SEN5A, either in the wild-type form or in the D1275N form, and driven by the cardiac-specific CMLC2 promoter. And on the other hand, as a reporter gene, enhanced GFP, driven by the eye-specific alpha-crystalline promoter. This construct was injected in single-cell zebrafish embryos, where it was integrated into the genome and transcribed and translated to protein. We generated stable transgenic lines by selecting for fish with green eyes, and then measured protein expression in those transgenic lines. And by Western blot, we quantified the amount of protein that was expressed to make sure that SEN5A wild-type and mutant lines would express similar amounts of protein. Now, to phenotypically evaluate these transgenic zebrafish, we did video microscopy, which allowed us to measure things like heart rate, contractility, heart rhythm, and so forth. And we consistently looked at 3 groups. The black group is the wild-type group, so these are non-transgenic fish. And then we had 2 transgenic lines, one expressing the wild-type human sodium channel and the other one expressing D1275N. As you see on the top here, the mean heart rate was reduced in D1275N transgenic zebrafish, but not across the board of all fish. When we looked at those fish that had a heart rate below 130, which we classify as bradycardia, then we found a significant increase in the proportion of fish in our mutant group in comparison to our control groups. And those mutant fish also had an increase in the occurrence of sinus pauses and atrioventricular conduction block. And that was concurrent with a decreased survival of that group. Now, to further investigate the phenotypic variability, we analyzed over 100 individual SEN5A transgenic fish over the course of the first 25 days of their life. Representative fish are shown here on the left. 5 fish in each group, 1 fish per row, were examined over the first 25 days and were examined on 7 different occasions. If on the day of examination the fish was normal, it's depicted or color-coded in green. If fish were bradycardic, they're red, sinus pauses are orange, and AV block is blue. And as you can see, while there were occasional abnormal fish in the control groups, both the incidence and the severity of cardiac phenotypes in our mutant group was much larger. This is summarized here on the right in the Kaplan-Meier graph, which shows that in total about 3/4 of D1275N mutation carriers showed an abnormal cardiac phenotype, but that phenotype was variable in any given fish at different time points. Now, to gain more detailed insight into the electric activity of zebrafish transgenic hearts, we established the technology to record ECGs from these tiny zebrafish embryos as well as adult zebrafish, and that is shown here on the left. As you can appreciate, zebrafish ECGs in their morphology are actually quite similar to human ECGs, and our D1275N mutant transgenic fish showed the same phenotypic features that we'd already seen in our video analysis, namely sinus pauses and atrioventricular conduction block. And when those ECG parameters were quantified, which is here on the right, then we saw evidence of cardiac conduction defects indicated by a decrease in heart rate and an increase in the duration of the QRS complex or the PR interval. Now, on this slide, I'm showing you examples of the kinds of video recordings that are used for phenotypic evaluation, with below the 2D representation of the wall motion of those hearts. And I do need my mouse to play this. Here it is. Now, on the left, you see a normal 2-chambered zebrafish heart, A indicating the atrium and V the ventricle. And this heart is beating regularly, versus on the other side here is a mutant transgenic fish which shows sinus pauses as indicated by these short arrests of cardiac motion, if you so like. Now, finally, I would like to guide you through a last experiment, which is quite complex, so I hope you'll stay with me. In our transgenic zebrafish, we're expressing human SCN5A, but those zebrafish also express endogenous cardiac sodium channels. Hence, we were interested in the consequence of human SCN5A expression in the absence of endogenous sodium channels. Now, to study this, we first looked at wild-type zebrafish, in which their endogenous sodium channels were knocked down using morpholinos. This was performed by another lab prior to us, and the results are shown here. So if endogenous sodium channels are knocked down, zebrafish morphins are slightly smaller, have a curved body, small eyes, pericardial edema, and severe heart dysmorphogenesis. Going back to our context of transgenic fish, as you can see, the severe sort of morphin phenotype caused by knockdown of endogenous sodium channels was rescued by the presence of human SCN5A, both in its wild-type form as well as the mutant form. However, this rescue was partial, and in comparison to the wild-type protein, the D1275N mutant was less efficient in rescuing this morphin phenotype. Now, in conclusion, we've successfully generated a transgenic zebrafish model of a human cardiac sodium channel mutation, and we've shown that transgenic zebrafish recapitulate many aspects of the human phenotype. The presence of human NaV1.5 partially rescues the loss of endogenous sodium channels, which indicates that basic pathways through which NaV1.5 acts are conserved. But this rescue was less efficient in the presence of mutant NaV1.5 in comparison with wild type, which indicates that mutant NaV1.5 is functionally compromised. Oops. The significance of this lies in the fact that this is the first proof of concept that stable transgenic expression of genes triggering cardiac arrhythmias in humans can provoke appropriate phenotypes in zebrafish. Our findings support the use of genetically modified zebrafish for differentiating functional from non-functional human gene variants, and we would like to propose that transgenic zebrafish models are powerful tools for evaluating pathophysiological mechanisms of human gene mutations and to develop and trial new therapies. I'd like to thank you, and particularly my host lab, Diane's lab, particularly Gunjan, who helped very much in this project, and also our collaborators in the Vandenberg lab, particularly Stefan, who helped with the electrophysiological side of things, our aquarium staff, and of course our funding sources, including the St. Vincent's Clinic Foundation. Thank you.
That was on time.